which is the movement of substances crossing the plasma membrane. So this chapter is quite important as well, because this is gonna be an important chapter which gonna be talking more of the function of the plasma membrane, because plasma membrane is covering the cells that we saw in animal and the plant. So in these cases, like, well, let's see the chapter outline. So we're gonna analyze what is the movement, how the substance are going in and out of the cell through the plasma membrane. And in daily life, there are a lot of things happening because of what substances are going in and out of the cells. And finally, we're going to see the movement, why is it important. So let's start with the structure of the plasma membrane. So we can see this is a plasma membrane structure. Okay. And let's see the structure before going through all the processes marked here. So like structure, basically, plasma membrane is made up of phospholipid molecule. So you can see this is the phospholipid molecule at the upper part, which is this is the external environment outside of the cell, and we have the cytoplasm inside. So these phospholipid molecule are arranged in this manner, okay, in and out, and since they are forming like a layer, which is like a bilayer, two layer, they are forming the phospholipid bilayer, as we can see here. The portion that is outside of the cells or inside the cells, this is called the oval part, is the head of the phospholipid, are called the head, and these head are called hydrophilic. Okay, they are hydrophilic because they are not scared of the water. So hydrophilic, so they can accept water, they are prone to water. So that's why they're outside. Comparable to the tail, okay, which is like branched into two here, they're called the hydrophobic. They're scared of water, so that's why they can't actually show themselves outside. Okay, so they are being inside in the bilayer. So let's see again. It's composed of phospholipid molecule, and since they are in a double, in a bilayer, so we call them phospholipid bilayer. And if the head are outside, they are okay with the water, so they're hydrophilic. If they are the tail are inside, because they are scared of water, so it's hydrophobic. Next, we have few other component and structure here. Okay, we have something called a proprotein. So proprotein is basically there's actually opening here, so substance can go in and out without any problem. Next, we have something called a carrier protein. Okay, this is also a carrier protein, and we're going to see what this carrier protein and this carrier protein are different as we go with these processes. So I hope you can see some of the differences of the structure, first of all. And next, we're going to move to the transport of the substances. So in here, we have the facilitated diffusion, simple diffusion. Okay, we have like osmosis, we have the active transport. So we're going to see the facilitated diffusion first. So we have two examples here. Some facilitated diffusion is called a facilitated because they require a protein. They are being held by a protein. In this case, it's a protein, or it can also be a carrier protein. So facilitated diffusion, it involves the help of a protein. It can be a protein or carrier protein. Protein can only carry a small inorganic ions, and osmosis like water can go in and out. So example of the ions is like sodium and potassium and also calcium. So all these items can go through protein without the need of carrier protein. On the other hand, like there are certain cases where protein is not sufficient because the substances are quite large. So in this case, we have large and soluble substances like glucose and also amino acids. So they can't go through pore, so they need the help of a carrier protein. Next, we're going to see the name says simple diffusion. So in the case of simple diffusion, they doesn't require help of any protein. So they're basically going to just involve coming in and out through these phospholipid bilayer. So all the lipid soluble, okay, molecules like fatty acids, glycerols, steroids are all fatty material, and also the vitamin that can soluble in the fats are A, D, E, K. All can go through this quite easily through the lipid bilayer. So we see facilitated diffusion, simple diffusion. Next is we also have another simple diffusion, which is another small molecules but they shouldn't be charged because the charge requires facilitated diffusion. So like oxygen, carbon, and also carbon dioxide all can go through this lipid bilayer, and water also can go through this and also go through facilitated diffusion. And the last will be the active transport. The name say active, so we will see the reason why is it called active in the later part of it. So in this case, they only carry certain molecules and iron. They have certain reason for that, which we'll see the example why are these called active transport. So overall, we have seen the structure of the plasma membrane, the example of transport. We will see it one by one later part, more in more detail. So first, we're going to see the facilitated diffusion in a more depth manner. So this is a facilitated diffusion, and we are focusing on the carrier protein here because pore protein is quite easy to understand. It's basically going in and out. So why we're going to focus on carrier protein? Because there are going to be certain processes, mechanism that taking place that allow these certain materials to be taken in and out of the cells. So in this case, let's see the mechanism, the process first. So this is outside of the cell, and this side is inside of the cell. So in outside of the cell, which is there's a lot of materials here. Example, if we have amino acids, we have glucose and all the small protein, and the cell require all these items. So these items need to be taken into the cytoplasm of a cell. So what it's going to do is all the materials, all the components will be actually first bind to what we call active site. So binding to the active site. Okay, this is a carrier protein, and it look like some kind of opening, but it's closed here. So first you need to bind first. So you can see the material have actually bind. So when already bind to the specific site, and later part, these carrier protein will now change shape. So first you can see the opening is quite big here. Now there's some opening, and which this changing of shape, the material which is like trapped here just now can just easily pass through. And because of that, the material is now in the cytoplasm, and after the process ended, it become to the initial shape, original shape of it. So let's see again, material outside need to be going into the cytoplasm of the cell. First, it will bind to the binding site. When it bind to the binding site, there will be some changes of the shape of the carrier protein, which the changes are going to allow it to go inside the cell. And once it's inside the cells, the carrier protein will go back to this original shape. So these are the mechanism that take place. It looked very simple in the previous diagram, but this is how it's gonna take. So attach, change shape, and release and come back to the original shape. So this is a facilitated diffusion through a carrier protein. So next, we're gonna see an example of the active transport. So as I said, I will explain further on the active transport here. So we have active transport. In this case, we have the material, which is the charged molecules. So we have all the sodium outside of the cells, and these are the inside of the cells. So in this case, we also require a carrier protein. So this is how the carrier protein for this sodium transport look like. So in case of this, active transport is because like there's a higher concentration outside of the cells, and all these need to be taken inside the cells. So first is all the sodium ion gonna first bind to the binding site, and this item when they want to go into the cell, that's we need to pay for the price we want to put all these sodium into the cell. So the price is actually paid by the use of energy. So we can see ATP is energy. Once it's released is phosphate, it become ADP. So splitting of this ATP will result in the sodium will be pumped inside the cell. So once the energy given for it for the sodium to be pumped inside, and the shape of the binding site will change, and basically we can see that the sodium is inside, and we have a lot of still sodium outside. So this process will be repeated again and again. So just recap again. So we have carrier protein and all the sodium outside the cell. First it will bind, and with the presence of the energy, we will pay using energy, and all the sodium will go into the cell, and the carrier protein will come back to this original shape. So since active transport, as we know, the word active saying that it require energy. So please remember, active transport require energy compared to simple diffusion or facilitated diffusion. So that's about the transport part of it. So next, we're going to see what happened if these processes are taking place into the important two cells of that we have learned in previous chapter, which is the animal cells and the plant cells. So first, we're going to look at the animal cells. So in here, we're going to see effect of different solution. So first, I'll introduce what is isotonic, hypertonic, and hypotonic. So isotonic is basically the solution where the water will go in and out of the cells at the equal rate. Where hypertonic is basically a concentrated solution and water will be going out of the cell. But hypertonic is less concentrated compared to hypertonic, and water will go into the cell. So in this case, we're going to see example of animal cells. Obviously, we are seeing red blood cells here. So because red blood cells is animal cell. So let's particularly look at the isotonic solution compartment here. So when we put red blood cells inside the isotonic solution, example in a beaker right now, water will go in and also water will leave at the equal rate, and this process is done by osmosis. So water go in to the cell, water will leave at the equal at the same time. So because of this water go in and out are the same, there's no problem with the shape of the animal cell. So the shape will retain as the normal as it should be. But in case of hypertonic solution, a very concentrated solution, when we put the red blood cells, water are going out, okay, continuously by osmosis. When there's too much of water going out of the cells finally become shrink, okay, it become like squeezed out of the water is being squeezed out. And in sense of red blood cells, we say the crenation had occur. And remember, crenation only can be used to refer to the red blood cells that have been shrinked. So other crenation for the red blood cells have occurred. So in case of hypertonic solution, water is diffusing into the cells by osmosis. Okay, because there's continuous water going in and in. As you can see, the red blood cells from a normal structure shape, it looked like bulged up, and because there's too much continuous water going in to a certain level, it's gonna finally burst. Okay, and finally when it bursts, we say hemolysis has occurred, and hemolysis is referring to breakdown of or rupture of red blood cells. Hemo means blood, so hemolysis have occurred. So just look back again. Okay, so in this case, isotonic, water go in and out, retain the shape. For hypertonic solution, water only go out and crenation happen, the cells shrink. And for hypotonic solution, water go in, and at one situation it's gonna burst, and in red blood cells hemolysis have occurred. So this is an example of how different solution on animal cells. Okay, let's see example on the plant cells. So we have a plant cells here. Okay, you can see there's vacuole, you can see there's chloroplast, and obviously we can see the cell wall. So again, I think you know what's the solution and what they're gonna do. So let's see the isotonic solution. So water going in to the cell and water going out the cell at the equal rate. So because of this, no problem. As water going in out at the same equal rates. So it's gonna the shape gonna retain the same. But in the case of hypertonic, water is continuously going out by osmosis. Okay, and because they have like this plasma membrane covering it, so this material basically we can say that the plant cell are not gonna basically shrink like animal cells. Okay, due to the vacuole and the cytoplasm can be shrink, you can see it shrink. Still, we have the cell wall. Okay, cell wall protecting it. Okay, because of this case, plasmolysis have occurred, and the plant become flaccid because water is being removed too much of the plant cell. So basically, it need water. Okay, become flaccid. You can see an example when you're not pouring water to the plant, and you can see the leaf is basically like no, not in in upright position. It's basically like bending down. Okay, so it's become flaccid. In case of hypertonic solution, water will be going in continuously by osmosis. Okay, and the water will actually get into the central vacuole. And too much of water going in will result in the vacuole become bigger and expand. Okay, and as it expand, it's going to basically push to the cell wall. Since there's a cell wall, there's no chance of it burst like animal cell. Okay, and this situation are now called the plant cell become turgid. That means the water are good inside the cells, and it's actually turgid. So let's recap again in plant cell. Water going in and out, the cell remain the same, the shape. In case of hypertonic solution, water going out. Okay, and in this case, plasmolysis happen, and you can see the shrinkage of this vacuole, and flaccid is happen. In the case of hypertonic solution, water are going in continuously. Okay, and it's going to push against the cell wall. Since we have the cell wall, there's no way of bursting, but the plant cells is said to be turgid in this case. So that's about this solution and different plant and animal cells. So let's look at the concept map here. So we saw the structure of plasma membrane, okay, which is called the phospholipid bilayer, and we have certain protein, carrier protein, protein is responsible for the movement of the substances. Okay, and we also saw the transports, which is passive. Passive means no energy. Active will require energy. It can be simple diffusion through phospholipid bilayer. Facilitated diffusion is require certain carrier protein, and we have osmosis where water going in and out of the cells. Okay, and we see that different materials are going out in the cell, and it's involved in everyday life processes. We also like learn about the concentration of solution. Solution concentration can be divided into isotonics. Okay, water in and out are the same, so the shape of the cell is the same. Hypertonic solution is less concentrated compared to hypertonic, so water will actually go into the cells. So when the water go into the plant cells, it become turgid or turgid. And in this case, when hypertonic solution, when we put red blood cells, water go in and resulting in burst of the cell, which is called hemolysis in red blood cells. Okay, and for the hypertonic solution, the coagulation is concentrated. So when we put the plant cell, so the water will be out of the cells and the plant will become a flaccid. In this case, it become plasmolysed. For animal cells, so like red blood cell we saw just now, so water is going out of cells. So at one stage, it becomes shrink, and this process is called crenation. So let's see some example of the question in chapter 3. So again, we are seeing all the phospholipid bilayer of the plasma membrane. So let's see the question. Name the structure label P. So let's locate where is P. P referring to the overall phospholipid bilayer. Okay, so obviously this is a phospholipid bilayer. We can see the head outside and the tail inside. So hydrophilic head, hydrophobic tail. So what we have Q here. So obviously we can see this is a carrier protein because there's a shape and there's no pore comparable to R. We can clearly see a pore. So in which side of the membrane, X or Y? We have X or Y is the cytoplasm is located. So obviously in this example, we can clearly see X is the outside of the cell because we have some carbohydrate that attach to the protein. So this carbohydrate, like a chain, can give some structure flexibility and structure support. So when we see the carbohydrate is at the branch outside, so X obviously is outside. And furthermore, we can see some kind of fiber. These are all called the cytoskeleton. So cytoskeleton making the cell flexible and allow this cell to be in a good shape here. So next question is like, state the function of cholesterol. So cholesterol is the one in middle here. Okay, red, yellow in color. So phospholipid bilayer, which have this cholesterol, going to make it stronger, more flexible. Next question is on the structure of R. So as I say, this is a carrier protein, and this is obviously a pore because you can see opening here. State the characteristic of substance that can cross R. So what material can go in and out of say this pore protein? Only small protein because large protein will go through carrier protein. Small protein can only go through this pore protein. So we have inorganic ions. So like ions can move through these R by facilitated diffusion using a pore protein. So they can have specific shape, only certain shape can go through this and to allow from outside to inside. So these are the example of question regarding the plasma membrane. So this is question of two different type of transport. We have A here and we have B here. So process X and process Y. So first we need to analyze the diagram given here. There are some clues here so that we know that what process is this. So extracellular, and this is inside the intracellular. Okay, similar to this B also. So let's see, they have phospholipid bilayer, both, and in this case we have some kind of protein. Okay, in this case it look like a carrier protein because when the material are going and the substance are going, there's a change of shape. You can see it's like closed here and it's opening here, and it's actually pumped into the cells. And in this case, there are certain carrier proteins also, and we see that all the materials coming in, and we see a lot of this PPP, and the clue is here where there's an ATP, the energy present here. So we already know this process quite early. So let's answer the question. So what is X? X is basically involving this carrier protein. So it's actually being facilitated. So you can write facilitated diffusion in this case. So why we've been seeing there's an ATP, an example here we have like carrier protein that's similar to a sodium pump. So that's obviously an active transport. Okay, so next is we're gonna see what differentiate these X and Y. What is the major differences we can see. So in case of facilitated diffusion, the molecule are moving from a region of higher concentration. So you can see the concentration outside the cell is quite high here, comparable to here. Okay, so facilitated diffusion, or most of the, we can say the passive diffusion is basically passive transport are basically movement of molecule from higher concentration to a lower concentration without energy requirement. On the other hand, why is an active transport? Because the material is like basically moved from the lower concentration. So you can see like in this case, example is 1. Okay. Mm, here. So it's like basically lower concentration, and we basically gonna increase the higher concentration. So in this case, we need an energy to move out of the cells. So we can see the type of substance example. So in the case of X, you can give glucose because they just need all this carrier protein for it to go into our cells. And in case of sodium ions, obviously require an active transport in the presence of an ATP energy. So these are the different questions you can see comparing two different type of diffusion and transport. So next is more of an application question. So you can see that there are two here. This is A and B. Okay, we have like substance P here and substance Q here. So name the process that is involved in movement of P and Q. Before we go further, let me explain what this diagram look like. So we can see alveolus. Alveolus is basically the place where the gas exchange going to take place. So gas going to go in, like oxygen going to go in and basically transfer or go through this process into the blood capillary, and we have the red blood cells are going to take all the oxygen into the blood capillary. And this obviously we already been given a clue. Okay, this is a root hair. Okay, we can see the root hair branching into the soil, and obviously all the materials are going to be taken into the root hair and into the cells and provide later to the plant. So let's see what the questions P and Q. So in this case, this oxygen example, which is the P material, okay, can gonna go through into the blood capillary as simple as passing through uh, this cell membrane by simple diffusion. So there's no requirement of energy. There's no requirement of any carrier protein or protein. So in case of root hair cells, we have a lot of nutrients in the soil. And since this nutrient is like, for example, you have sodium inside the soil, all this sodium need to be taken by the root hair cell for the plant usage. And obviously, if it comes to sodium, it's about active transport. So further, the question asks, please explain how this process is happening. So we're going to just refer to the previous diagram that we saw about active transport mechanism. So sodium will bind to the carrier protein. In the presence of energy, they're going to be change of shape of the carrier protein, and the sodium going to be pumped into the root hair cells, and then the cells going to take the sodium. So that's how as simple as we can see it. So this is another question on the solution concentration. So here we can see that this is a plant cell, and the plant cells are being in a type of solution. So we do not know, the question is asking that the reason what the solution look like, because the cell before is like this, and later when we put into the solution, it turned out to be like this. So explain the condition. So the cell is actually to see being flaccid, okay? There's less of water here because water is going out of the cell, okay? It's being like shrink. And when we put into another solution, it turned out to be a normal cell's bag. So in this case, obviously water have gone into the cell. So water going into the cell. So this solution is probably a hypertonic solution. So after being immersed into solution and hypertonic solution, the water go into the cell, and obviously water go into by process of osmosis, and more water going into the vacuole. So the vacuole expanded and enlarged, and cytoplasm is being pressed against the cell wall. So water, even though going in, it's not going to burst the cell because we have the cell wall. And then the cell is now said to be turgid. So these are some of the example in chapter 3 that we have like seen